we were talking about ribosomes, the nucleus, ribosomes, making proteins, and then I brought up the endoplasmic reticulum. We have what we call the endomembrane system, and it's going to consist of three parts. I'm just giving you a heads up. The endomembrane system is responsible for secretion of materials outside of a cell, and it's the first of the parts that it's going to consist of is what we call the rough endoplasmic reticulum. So we said if you are a protein that is going to be secreted to the outside, you will not be made on a free ribosome. You will be made on a ribosome that is attached to the endoplasmic reticulum. Now, when scientists looked at micrographs of the endoplasmic reticulum, some of it looked rough. They said, whoa, that's rough. What are those little dots? And then they figured out those little dots that made it look rough were ribosomes. And so the portion of the endoplasmic reticulum that's closest to the nucleus is where you're going to have the ribosomes. So the messenger RNA comes out, finds a ribosome on the surface of the rough endoplasmic reticulum. It makes that protein and it puts it inside of that system of channels. Now that system of channels is continuous with, connected to the smooth endoplasmic reticulum. The smooth endoplasmic reticulum, it does not have ribosomes, but it's continuous with it. And it is going to be where the protein that was made on the rough endoplasmic reticulum is going to make its way into the smooth endoplasmic reticulum. And what's going to happen is that smooth endoplasmic reticulum is going to pinch off a little ball with that protein in it and send it to the third part of the endomembrane system, known as the Golgi apparatus. Okay, so the smooth endoplasmic reticulum, it's sort of a passageway, and it also makes phospholipid bilayer. So if you think about it, if you're always pinching off little balls of phospholipid bilayer to send them to the Golgi, you're going to be gone, right? You know, you have to keep making more of that stuff. So that's one of the things that's going on in the smooth endoplasmic reticulum. If you are a cell in the liver, we'll say, or kidneys that does detoxification, you have enzymes in there that are helping out with that as well. And in some cases, making of lipids for secretion, like testosterone, the steroid hormone, that would happen in the smooth endoplasmic reticulum as well. So we have this system of tubes that is separate from another system of tubes, which is just towards the outside from there, and that is called the Golgi apparatus. And so we're following the path of this secretion product to the outside. So when we go to the Golgi apparatus, you can see in our picture there, we have a blue nucleus and then a red endoplasmic reticulum and then a purple Golgi. And all the way at the bottom left corner down there, you can see a little bit of the cell membrane. And so we had said, what's going to happen is that smooth endoplasmic reticulum is going to pinch off a little ball. It's like a soap bubble. Remember, we said it's like soap bubbles. And that thing's going to go bubble, 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 bubble across and join up with the Golgi. And so it transports the products from the rough endoplasmic reticulum through the smooth endoplasmic reticulum, where it is sent in a vesicle to the Golgi apparatus. Now the Golgi, what we said you need to know the job of each of these. What is the job of the Golgi? The Golgi is in charge of vesicle traffic. It decides when it's going to send that that chemical to the outside. So the Golgi brings it in there and holds it there, and then it'll package it up in another one of these transport vesicles, and it'll bubble across to the cell membrane, join together with it, and release it to the outside. So the Golgi, I'm just going to look at my notes or my slide here real quick. It says it packages secretions for exocytosis, release to the outside, renewal of cell membrane. So remember we said that phospholipid from the smooth endoplasmic reticulum, it got transferred to the Golgi. Well, what happens? The Golgi then packages it up and transports it to the outside. Hang on, please. To the cell membrane, and from there it gets released to the outside when the Golgi is ready to turn it loose. I have somebody at my door, so I better stop right now.